uh, start. Oh, actually, let's pray, and then we can get started. So it looks like the main thing I'm praying for is the tree on Anna's house.
Yep, it's big.
So are we praying for the tree or for the house? I'm just kidding. I'm just kidding. Right. So we'll pray that the damage that you guys can take care of that in a timely manner. Are you living in the house?
No, we're staying at my parents' house right now, which
Okay.
is always a blast.
Yeah. Just if it makes you feel any better, it doesn't get any better as you get older.
Oh, yay.
So okay, so we'll pray. We'll pray for that. We'll continue to pray for everybody that's been affected by the severe weather that kind of ripped through the southeast. Anything else in particular before I get going here? Okay, cool. Let's pray and then we'll get started. Lord, just thank you for this day, this opportunity to be here with one another. Ask that you bless our time together as we gather in this virtual platform. Today, just specifically lift up anyone that's been affected by the severe weather. In particular, our good friend Anna, who currently has a tree on top of her house, and just ask that that whole process of removing the tree, of repairing her and Jeff's house would go as smoothly as it can. And we just ask that you show them favor as they deal with contractors, construction workers, insurance agents, that would all resolve as quickly as possible. We also thank you that queso and what's the other one?
Pico.
Pico. I knew it was some other type of food. Queso and pico are okay as they were in the house when the tree fell. And I just ask that you continue to be with those that lost anything, whether it would be loved ones, material things in this storm, and that you bring them peace and comfort as they try to put their lives back together. I also continue to lift up anybody that's dealing with COVID. I pray for healing for their bodies. I pray for protection for those that are healthy and well. And I pray for wisdom for our leaders and our healthcare workers as they determine how best to proceed forward and what precautions need to be taken and what precautions can be eased. And I also just continue to lift up any unspoken prayer requests around these students' hearts. Thank you for loving them and hearing them. It's in your son's name that we pray. Amen. All right, so here's the deal, and I mentioned this again, and I will mention it this time, and I'm also recording this so that I have proof that I reminded you of this. You guys saw that the emergency grading scale has been enacted, and the way that works is at the end of the semester, when you see your final grade, you can decide, hey, I want my letter grade, or I would rather take a P, right, which would be a D and better, right? An F is still an F. Now, the kind of caveat or the deal with that is I get to set the attendance policy for the rest of the semester. Yay me, there's only like a week and a half left, so the power does not feel that great. And so my kind of deal for you guys is that, between now and the last class, you can be out one class, okay? I'm hoping that you are not out any classes, all right? But you can be out one class. Now, again, I understand if there are some severe internet issues and things like that, especially if you're in this area because of tornado, then you need to reach out to me and let me know what's going on. But let's say you miss 2 of our next couple classes and you have a D and you come to me and you're like, I want to go for the pass instead of the letter grade, and I will say no. All right. So keep that in mind, right? There's a kind of a stipulation that goes with that. Those of you that are nursing, I can't remember who asked last week. I sent an email to Jake asking him about how that affects you in the nursing program. I've not heard back from him yet. So when I do hear back from him, I will shoot you guys an email. The good news is you don't have to make any of those decisions until after grades are in from the final. All right. So just
They actually sent us like a to all the nursing majors. I think they sent us all like one of those kind of deals. So
they
he
did.
might not. Yeah.
Okay. Can you forward that to me just so I can see it?
Yeah.
Okay. Cool. Thanks, Emily. She's my narc in the School of Nursing. All right. With that being said, let's move forward. Now, my plan for you guys, so this was quiz 10. So I'm just going to tell you right now, there is going to be a quiz on Monday. And then there will be a quiz on the following Monday. So the next 2 Mondays, you guys will have quizzes that will put you at 12 quizzes and allow you to replace 2 of your quizzes. Okay. And I'll try to remember to send out maybe an email with all of this. I'm also just playing with the idea of, we'll call it a COVID-19 extra credit, where I give you guys points that would have been probably earned doing the extra credit games. And so I think I'm probably playing with the idea of assuming that all of you got first place on your team and you would get those first place points. Okay, so be on the lookout for that. With that being said, let's finish up chapter 10 and then jump into chapter 11. Olivia, yes, your exam is next Friday. Oh, Natalie also asked that too, right? So for your lab, all right, lab exams will start next Tuesday. Unless you're in Riley's crew, right? Then I think, and Riley, you can correct me if I'm wrong. Do you know, are you guys doing your last lab exam the Monday of reading day week? Does that sound right?
I have no idea. We haven't really.
Okay, perfect. Riley, could you do me a huge favor and check into that for me before Friday? Just so I know, you're my inside source. All right.
I will email her right now.
Okay. And I mean, and I just figured that'd be good for you to know too. So we'll find out from Riley. Fortunately, I've made him the representative. Maybe unfortunately for him, he has a little extra burden of responsibility, but I believe he can handle it, right? But so yeah, lab exams will start on Tuesday for my crew. And then if you have me on Thursday, it'll be at Thursday. I'm assuming if you have Hobbs on Wednesday, you'll have him then. Perry on, I guess, Friday. I guess you guys get kind of hit a little harder. But lab will be in lab. Lecture will be in lecture. So you'll have lecture in here with me. I will open it at 11, and it will close down. I'm probably going to actually give it to like 12:05 this time. All right? Cool. Any questions, comments, concerns about that? All right, well, let's jump into chapter 10 and finish this up. There we go. All right, so if you remember, we finished just talking briefly about some of the different neurotransmitters, the different disorders that an imbalance in them can cause, and then some of the drugs, which can be good, that help, and some of them that can be bad. All right. Really, really briefly, I wanted to mention this idea of neuropeptides. Now, I hope this doesn't confuse anybody. If it does, let me know, right? Neuropeptides can act as neurotransmitters, right? Not all of them do, but they can act as neurotransmitters, all right? And another way to look at them or think of them is as neuromodulators. And so they are able to alter the response of neurotransmitters by either blocking their release. Alter the response to neurotransmitters. Okay, so they can alter the response, right? They can cause variations in that response, or they can just completely block their release. All right. Two examples of neuropeptides are enkephalins and beta endorphins, and these are produced in response to pain. Interestingly, they actually bind to the same receptor as morphine. All right. And so morphine, opium, right, all come from the poppy plant. And we've known these to be narcotics for a while. It was discovered, I think, in the 60s or 70s. And it was a really interesting discovery when they followed basically the morphine in someone's system and saw that it was binding to receptors on their cells. And this was considered a really unique kind of discovery because morphine's derived from plants. And so we have a receptor on our cells that responds to something that's made in a plant. And it turns out, right, that occurs because we have essentially natural morphine. Now, what's really interesting is that these drugs or these natural neuropeptides prevent something called substance P from binding to these same receptors. Substance P is one of the reasons that you feel pain, right? So this helps block substance P and helps block pain. If you've known someone or know of someone that is a drug addict, one of the things that is very challenging during their detox, right, is that when they are taking drugs that mimic things like beta endorphins, their body actually stops producing beta endorphins because it doesn't think that it needs them because the system is flooded with them. When those patients come off of whatever drug they've been taking for a prolonged period, they do experience high levels of pain because once the drug's removed and their body doesn't realize that it needs to start producing its own beta endorphins, substance P can bind to these pain receptors without being blocked. All right, so that's one of the challenges of detox. All right. The last thing that we're going to talk about in this chapter is this idea of impulse processing and neuronal pools. All right, so a neuronal pool is a group or a collection of neurons that have that are functioning together, right? That are trying to carry out the same process. Now, it does not mean that they have to be in the same anatomical location, right? Because axons are far-reaching, right? You could have a neuron that's up in the brain that is part of the same neuronal pool of a neuron that is in the spinal cord, right? They're contributing to the same function, right? They are signaling to one another. They are able to stimulate one another, and so we consider them in the same neuronal pool. When we talk about neuronal pools, some of the different kind of impulse processing that we have going on is something called convergence and divergence, all right? And so you can probably look at these and guess which is which, but when we talk about convergence, you see that right here. So you have neuron 1 and 2, their axons are extending down to neuron 3, all right? And so this could be really important for generating or helping you reach a threshold potential, right? Causing enough of a depolarization event. So if you just had one, it might not happen. This convergence also allows neurons to integrate information from other neurons and begins the process of taking all that information together to generate some type of impulse. Right? You also have divergence, and in this case, right, you see an axon that branches, and that axon is hitting two different neurons. This is actually really important because it can lead to signal amplification. This idea, right, that one neuron, right, can signal to two neurons, right, so you amplify that response, right, one neuron affecting two neurons. Theoretically, down here, right, you could see this even diverging and then affecting more neurons. One thing I want to point out is that it shows these here. It looks like they're kind of exclusive events. This is not the case, right? You could have convergence and divergence going on all within the same neuronal pool, right? It simply has separated them here to kind of point out to you or indicate what one looks like, right? Because if you put it together, it'd be kind of dicey. One other thing that I want to point out before we leave this chapter, right, is that when we talk about the strength of an impulse, and I'm sorry I haven't mentioned this before, but what I want you to realize is that an action potential is an action potential. The strength of an impulse is actually determined by how often that impulse is moving down a neuron, right? So if an impulse is coming every 10 seconds down a neuron, it is not as strong of an impulse that is coming down that axon or that neuron every 0.1 seconds, right? So it's a timing thing. The quicker those impulses are together, right, the stronger the signal is. Got one chat here, let's see. We have one more Monday lab and then we'll have the nervous. Okay. Yeah. So thank you, Riley, for that information. Sorry for the quick interruption here. But yeah, so if you are in Riley's crew in the Monday lab, your last lab exam will be a week from this upcoming Monday, right? So that'll be the week of reading day. Okay, cool. Thank you, Riley. All right. So now let's actually start to talk about more of the anatomical division of the nervous system. All right. So this is moving into chapter 11. Where did it go? That's interesting. Hold on. Being weird. Okay, I gotta exit out of this. There we go. Okay. All right, guys, sorry, I'm having a little bit of a technical malfunction here. File, close, okay, close it. Again. Okay, let's see if I fixed it. Maybe I did. Ha! I knew I could beat technology. There we go. All right. Divisions of the nervous system. So again, we will still talk about physiology here, but we're also going to focus a lot more on the anatomy and the structure. And this kind of ties in nicely, or some of it ties in nicely to the last lab of the nervous system. So those of you that have had Tuesday lab and those of you that will have lab today, this will be kind of a little bit of a refresher, some parts of it. Those of you that haven't had lab yet, this will help reinforce what you see in lab this upcoming, or these upcoming next couple of days. Let's talk about division of the nervous system. So one of the first things we're going to talk about are ventricles and cerebral spinal fluid, right, CSF. Within the central nervous system, right, when we talk about the brain, you have 4 ventricles that are contributing to the production of the cerebral spinal fluid. You can see on each hemisphere of the brain, you have 2 lateral ventricles. It's actually the left ventricle that's considered ventricle number 1, the right ventricle number 2 for those lateral ventricles. It's kind of hard to see here, right? But here's the 3rd ventricle. If you go back and watch the dissection video from Professor Dorset, or when you have lab, pay attention if you haven't had it yet to that dissection video that you will watch from Professor Dorset, and you'll see that this 3rd ventricle sits right atop the corpus callosum, right? The corpus callosum is what joins the 2 hemispheres of the brain. And then you do have a 4th ventricle that is going to sit just above the cerebellum. And I think we'll see what that looks like again from another view. All right, so here's just showing you a lateral view of the brain so you can see, again, kind of how big that lateral ventricle is. All right, so these ventricles are producing cerebral spinal fluid. And here, this is just giving you kind of another view. So you have, let's see, so here's showing you where that fourth ventricle is. So here's the cerebellum, all right? So it's kind of in front of the cerebellum, and you can follow the arrows that's showing you the flow or the direction of the cerebral spinal fluid. Another thing I want to point out, and I'll mention this again, but here you have things called choroid plexus of the fourth ventricle. Here's choroid plexus of the third ventricle. There are choroid plexus present in the two lateral ventricles. These are extensions of one of the meningeal layers, the pia mater, which we'll talk about here in just a second again. But these extensions of the pia mater go into the ventricles. They're made up of, or they contain those ependymal cells that we talked about that are producing cerebral spinal fluid that then circulates around the whole nervous system. And for those of you that have had lab, remember one of the things we pointed out in the spinal cord is the central canal, and cerebral spinal fluid is running through the central canal as well as surrounding, all right, the central nervous system. And those other layers of the meninges, here's the dura mater, right? That's that tough outer layer that provides protection. This is also where blood vessels enter into the central nervous system, and there's a lot of nerves that are running through it also. You have the arachnoid mater, which is really important again is important for the production of cerebral spinal fluid along with the choroid plexus of the pia mater, right? So here, if nothing else, right, please appreciate that the central nervous system essentially is being bathed in cerebral spinal fluid. Now, why is the cerebral spinal fluid important, and how do we actually use it as a diagnostic tool? So let's start with the first one. Cerebral spinal fluid, because it surrounds the entire central nervous system. Technically, right, your brain and your spinal cord are floating in CSF, all right? And it's important that they float in CSF because that actually means or that extra fluid that allows them to float provides a little bit of cushion, right? So when you get a bump on the head or you get hit in the back, right, it typically is not a big deal. Now, if you've had a concussion, right, a kind of general way to view a concussion essentially is as a brain bruise. And so your brain essentially hits off the inside of your skull, right? So the force of impact is more than the cushion of the cerebral spinal fluid can absorb. But for day-to-day wear and tear, right, it provides enough cushion so that every time you move your head or do something, right, you're not getting a concussion. So it's really important there. As far as a diagnostic tool, all right, we can use this for spinal taps, and spinal taps can tell us a couple of different things. One, right, we can pull cerebral spinal fluid, and you can see here they're pulling it from this subarachnoid space, so that's what is filled up with the cerebral spinal fluid. You can pull this out and you can determine if the patient has meningitis. You can also determine if they have a severe brain injury or spinal cord injury that's causing bleeding, all right? If there is blood detected in the cerebral spinal fluid, that means you have some type of hemorrhage going on within the central nervous system. This can be really bad because as that blood starts to hemorrhage and clot, it can put pressure on the central nervous system, which can damage the tissue, and depending on when it's addressed, could lead to things as severe as death, all right? One of the things I want to point out here, right, is that the spinal cord, right, is right here. It ends, right, as far as being, I mean, there are nerves that extend off of it, but the actual structure of the spinal cord, you can see here, does not go past, or I think past the second lumbar. I have to go back and double check and make sure that's what that is because I can't see all of it. The second lumbar vertebrae. And so when you do a spinal tap, right, you do it farther down the lumbar vertebrae, a little bit closer to the sacrum. And here, when you go in with a needle, right, you're not risking any damage to the spinal cord. And I think Maddie had asked maybe on Monday about epidurals. So this is where they would put an epidural in, is approximately the same spot. And so an epidural, right, would then release nerve blockers into this space, but you're not risking actual damage to the spinal cord. All right. So that's a good question. I know, I don't know. Todd's medical expert sometimes is more than mine, so he can back me up here. For people that have, meningitis, you typically expect them to have fever, severe headaches. They could be experiencing some type bouts of paralysis, loss of vision, but usually a fever and severe headaches are going to be, one of the signs. So theoretically be seeing your primary or an ER doc, and when they heard some of those symptoms, they would
Yeah.
put you in.
Fever, headache, and blurred vision. Those are like your three main things.
Right.
So you got it.
And so, to remember for meningitis, there's two types, right? There's bacterial and there's viral. And most of you hopefully have been vaccinated for Haemophilus influenza type B, which is the main cause of bacterial meningitis. Viral meningitis, unfortunately, there's not much they can do. Kind of fortunately, it's typically not as severe, and you have to clear that like any other viral infection, right? But if not treated, right, you can have paralysis. You could have permanent loss of vision, and severe cases of meningitis can even lead to death, all right? Just kind of a quick note about ventricles. You can see this, this top image here. These are normal ventricles, and the ventricles are this size, or when you see them this size, it's because they're draining properly, all right? Here you have ventricles that are swollen, and the reason they're swelling is because for whatever reason, this patient is unable to properly drain cerebral spinal fluid. And so you can see that's not good because, again, if the ventricle swell, then they press on brain tissue, and that is an issue. So what you can do here is you can have a shunt put in, and the shunt actually runs from the ventricles down into the small intestine, where it dumps cerebral spinal fluid that can either be excreted or can be reabsorbed by the body. All right. That's just kind of a little bit about that. So let's start to talk about the spinal cord structure here. And you guys know this is one of the two main components of the central nervous system. Here you can see that the spinal cord, right, connects to the brain via the brain stem, right? And that connection point is through the foramen magnum. You guys hopefully remember that from the skull, right? It's a big hole on the inferior or underside of the skull, okay? And here, just kind of a thing that I want to point out, and you see this again, is that the spinal cord, right, has a cervical enlargement, right? So around the cervical vertebrae, it's a little thicker. You then also have a lumbar enlargement as you near the end of the spinal cord, right? So it's not just one continuous uniform shaped collection of tissue. And so this kind of just gives you a little bit of a different view of it. The spinal cord can actually be divided into 31 segments. And from these 31 segments, you have nerves that run off and are part of the peripheral nervous system, right? So these nerves that come off of the spinal cord form your peripheral nervous system, all right? And just kind of basic, right, regions, right? So this region of cervical enlargement, right, is supplying the nerves to your neck and your upper limbs, all right? And so anatomically, it kind of makes sense. For the most part, the areas of the body that are closest to that part of the spinal cord, right, are being supplied with nerves from that section of the spinal cord, right? The lumbar region, right, is supplying nerves for your lower limbs, all right? So it kind of makes sense. Here you have this conus medullaris, and this feeds off into some spinal nerves that will extend into the lumbar and the sacral regions. Now, again, remember the spinal cord is no longer, right, extending past about the second lumbar vertebrae, but there are still nerves that are coming off of it that are extending through the vertebral column before they leave that area, right? And then finally, you have this phylum or phylum terminal, right? And this is basically connective tissue, all right, that is helping kind of hold the conus medullaris, this last part of the spinal cord, in place by attaching, right, kind of serving as an anchor point or attachment point to the coccyx. Okay. Oh, now this image, if you, well, okay, let me say this. If you've had lab, no, you've all had the nervous system lab now. Yes, okay, good. So this is an image that you should be really familiar with, right? You will see this again on your lab exam. I'm not hiding that piece of information for you, all right? What we talked about in lab, right, were some of these different structural components. How do you tell the anterior from the posterior? Well, here's the anterior median fissure. Notice the two hemispheres. There's more space between them than the posterior median sulcus. Also notice gray matter from the posterior horn, right, extends to the dorsal root ganglia. You don't see that here with the anterior horns, right? And then remember the difference between the white and the gray matter, all right? So you have white matter that makes up the funiculi of the spinal cord, gray matter that makes up the horns. And what we're going to talk about here coming up in just a little bit are reflex arcs, right? And we'll even go over specific tracts that are ascending and descending through the spinal cord, right? So please know these different regions. You will see that again on the lab portion of your exam, and maybe even a small portion on the lecture. So here, my next minute or so that's left, I want to introduce the idea of reflex arcs, and then we'll talk some more specifics on Friday. All right. So a reflex arc, right, is an impulse, right, that we can follow along a nerve pathway. Right. Now this impulse, right, we care about the afferent, right, which is the sense, right? So this is a sensory impulse, the impulse that's gathered information, right? It's going to take that to the central nervous system. Sometimes that's all the way up to the brain. Sometimes that's just the spinal cord. And then we have an efferent or a motor neuron, right, that generates a response based on that sense, all right? Again, reflex arcs that are involving just the spine, we consider those simple or less complex, right? Because it does not take as much processing power or time, right? So this will be the last slide we talk about really briefly, right? I know this looks super simple, but it is important to kind of use this as the basis of understanding a reflex arc. So you have a receptor, right? In this case, a sensory neuron. It sends information to the central nervous system. This could be the brain and the spinal cord. This could be just the spinal cord, right? And then we have an effect. So right here you see there's a receptor, a cutaneous receptor, right? A signal is generated, right? That goes into the spinal cord. We'll talk about what an interneuron is or why that matters in a reflex arc in just a little bit, all right? That information is quickly processed. An impulse is sent out, all right? In this case, to an effector, which will be a muscle or a gland. All right, so that's the basics of a reflex arc. We'll talk about 3 different reflex arcs that exist within your body starting on Friday. All right, please remember there will be another quiz on Monday and then the Monday following that. I'm just trying to get you guys a little bit of extra points. I'll send out some more emails about what that kind of looks like. Ooh, and actually, because I just see, well, I'll open it up, but I see Todd right now on my screen. This one's for you, Todd. All of the Chem 21, right, that you haven't done, every single one is open until next Friday, all right? Every single one is open until next Friday. If there are any that you don't do or that you've missed by then, right, I'm sorry, I tried. Most of you are pretty good. There are a few that I hope take advantage, really take advantage of this, but they are open until next Friday. So please do that. Chem 21 open until next Friday. All right. Any questions, comments, concerns before I let you guys go? So I'm a little bit over. All right.
I'll send you an email, but yeah, the lab scores are still not in.
Okay. You just send me an email too, and I might even kind of forward that Dana, all right, and figure out what's going on. Or Professor Perry, sorry. All right, cool. I hope you guys have a great rest of your day.